Infectious Diseases, Preventive Medicine and Clinical Species Conferences 3 2024-2025. Fish, Crustaceans, Molluscs, Amphibians and Insect Diseases, CURS 8. Second part. Aquaculture is the fastest growing sector of global agriculture. The most significant sector of aquaculture production from an economic standpoint is shrimp farming. The annual value of marine shrimp production is estimated to be $40 billion in the global market, FAO, 2016. Over the past 3 decades, viral and bacterial diseases have become obstacles to shrimp farming, and many of these pathogens have spread worldwide. The resulting pandemics have collectively cost the global shrimp aquaculture industry billions of dollars. Of more than 30 microorganisms currently known to infect penid shrimp, 8 viral and bacterial diseases dominate the list in terms of their adverse effects and have been listed as notifiable diseases to the World Organization for Animal Health. The aquaculture industry's responses to threats of aquatic diseases include the development of emergency plans for specific diseases. Among these notifiable diseases to the WA is infectious myonecrosis, IMNV, caused by the infectious myonecrosis virus, IMNV. IMNV infects penid shrimp, including the most commonly farmed species, the Pacific white shrimp, and the black tiger shrimp. IMNV is classified as a member of the Totiviridae family and has been found to be most closely related to a virus infecting a flagellate parasite, Giardia lamblia, mammals, and birds. Thanks to cryomicroscopy and 3D imaging reconstruction using purified IMN virions, there are protruding fibers that form fivefold axes. These protrusions may play a role in facilitating extracellular transmission and pathogenesis of IMNV. The complete genomes of IMNV types originating in Brazil and Indonesia were sequenced and found to be 99.6% identical at the nucleotide level. The complete identity of the genome sequence at 99.6% indicates that the disease was introduced from Brazil to Indonesia in 2006. Transmission electron micrograph of infectious myonecrosis virus. Cesium chloride gradient fraction of purified virions. Stained with 2% phosphotungstic acid. Sensitive species. Species that meet the listing criteria as susceptible to IMNV infection includes brown tiger shrimp, banana shrimp, white-leg shrimp, and giant tiger shrimp. Species for which there is incomplete evidence of sensitivity include the blue shrimp and the southern brown shrimp. Global distribution. IMN was first reported in populations of P. vaname cultured on a shrimp farm in the state of Piauí, Brazil in September 2002. IMNV was first detected in Indonesia in early and has been reported from the islands of Java, Sumatra, Bangka, Borneo, Sulawesi, Bali, Lombok, and Sumbawa. IMNV has been detected in the West Bengal state in India in July 2016 and spread to Andhra Pradesh state, the largest livestock region of P. vaname in India, in April 2017. The most recent case occurred in Malacca state, Malaysia, in June 2018. Persistence in the environment. No data are available regarding the persistence of IMNV in pond water and sediment or on farm equipment. Modes of transmission. IMNV can be transmitted both horizontally and vertically. From oral bioassays, it is clear that individuals can become infected by ingesting infected tissues. It is likely, although there is no definitive evidence, that the virus can be transmitted through water or sediment, as has been demonstrated with other shrimp viruses. IMNV can be transmitted by ingestion of infected tissue, through cannibalism of sick and dead shrimp. Cannibalistic transmission is supported by feeding trials in which groups of shrimp ingesting infected tissues rapidly developed IMNV infection and suffered deaths. Shrimp larvae can be infected through vertical transmission. IMNV has been detected in the eggs and ovaries of infected females, suggesting that maternal transmission is involved. Vectors and reservoir hosts. The brine shrimp Artemia sp. is an essential food used in the larva culture and maturation of shrimp. Through laboratory infections, A. franciscana can act as a mechanical vector for IMNV. IMNV is an unenveloped virion that can probably remain infectious in the gut and feces of seabirds that feed on dead or dying shrimp on farms with IMN epizootics. IMN may be spread within and between farms through bird droppings or regurgitated shrimp carcasses, similar to one of the routes of transmission of VST. Native wild penaeid shrimp, including P. subtilis, in northeastern Brazil have been anecdotally reported as hosts. Factors influencing disease transmission and expression. The incidence of IMN generally becomes more frequent during the rainy season in northeastern Brazil. In laboratory infection studies, it has been shown that reducing salinity from 35 parts per trillion, normal condition, to 5 parts per trillion, stress condition, reduces the generation time of IMNV from 57.4 to 25.2 minutes, thereby increasing virus proliferation. This change may be related to osmotic effects on the shrimp's immune defense system. Impact of the disease. Mortality due to IMNV infection can be as high as 70% at harvest, with more than 40% mortality occurring late in the production cycle and reduced market value of survivors with substantially necrotic caudal muscles. Economic losses during the 2002 to 2003 NMPI outbreaks in Brazil were estimated at $20 million. Brazilian shrimp farmers suffered economic losses of $440 million as a result of IMNV outbreaks in late 2005. The accumulated losses of Brazil and Indonesia due to the IMNV from 2002 to 2011 have been estimated at more than $1 billion. Additional costs, not yet quantified, would include the costs of sanitizing facilities, implementing control measures, and tracking inventory. Clinical signs. Shrimp in the acute phase of IMN exhibit focal to extensive white necrotic areas in striated, skeletal, muscles, figure A, particularly of the distal abdominal segments and caudal fan, which redden after death, similar to the color of cooked shrimp, figure B. The distinctive clinical sign, whitish, opaque, muscular, is generally more prominent in P. vannamei than in P. stylirostris or P. monodon. In addition, the presence of these clinical conditions is not specific to IMN and therefore cannot be used for a definitive diagnosis. IMN can occur at any stage of shrimp grow-out and is a chronic infection. Mortality can reach 70% at the end of the crop cycle. Evidence of the disease, other than the appearance of a whitish tail muscle, is that shrimp become lethargic and have a reduced food intake. Signs of infectious myonecrosis. A. Red penas vannamei from an epidemic in Brazil, with varying degrees of skeletal muscle necrosis visible as an opaque, whitish discoloration of the abdomen. B. Bread P. vannamei from an outbreak in Indonesia, showing reddened necrotic tails. Mass mortality of penas vannamei in culture caused by infectious myonecrosis on farms located in A. northeastern Brazil in 2002. B. East Java, Indonesia in 2006. For histopathology, whole moribund shrimp or samples of their muscle tissue may be used. Samples should be stored in Davidson's AFA or an RNA-compatible fixer, pH 6 to 7. This is to reduce RNA degradation for ISH analyses, processed into paraffin blocks and sectioned. The tissue sections should then be stained with Handa using a standard protocol. The colored sections are then examined using a compound microscope. Infected tissues show lesions formed by coagulating muscle necrosis and hemocyte infiltration, figure A. Basophil inclusion bodies are observed in the cytoplasm of skeletal muscle cells, figure B, lymphoid organs, hindgut, and phagocytic cells in the hepatopancreas and heart. In addition, spheroids are usually seen in the lymphoid organ, figure C. Some are ectopic in the heart, antennal gland, and loose connective tissues, figure D. Although histopathology can provide a presumptive diagnosis of infection with IMNV, transmission electron microscopy, TEM, or specific molecular tests such as RT-PCR, RT-qPCR, or ISH, or immunohistochemistry, IHC, or Western blot analyses that detect viral proteins, are needed for confirmation. The diagnosis of IMNV in shrimp is usually made via a sequence of procedures after a preliminary diagnosis based on macroscopic clinical signs. These include histological demonstration of lesions, which is usually too slow to allow shrimp farmers to make practical management decisions. Molecular diagnostic methods are much faster and can provide immediate information to shrimp farmers. Molecular detection of IMNV has been used successfully, with RT-PCR being widely used for rapid and specific detection of the virus. On-site rapid diagnostic tests. Optimal on-site detection systems must be fast, sensitive, inexpensive, and easy to maintain and use by non-specialists. In addition, reagents should be supplied in a format that allows for easy shipping and storage. There are a range of different technologies currently available for rapid on-site diagnosis of IMNV. Reverse transcription loop-mediated isothermal amplification, RT-LAMP, combined with a lateral flow test strip, LFT, has been developed. The total analysis interval is less than 75 minutes. These dosages are carried out with a simple water bath and are inexpensive. A dipstick test, lateral flow immunochromatographic assay, UFIA, using 2 monoclonal antibodies to detect MCP-IMNV was developed. This immunoassay is not as sensitive as PCR-based methods, but it has the advantages of lower cost and speed. It gives results within 15 minutes of sample preparation, and is simple in format, not requiring expensive equipment or specialized skills. Thus, it is suitable for use by farmers to confirm the presence of IMNV. A commercial IMNV detection assay, based on isolated isothermal polymerase chain reaction, is also available for on-site detection of IMNV. These types of rapid diagnostic tests need to be evaluated by local field tests, after which their use can then be incorporated into national emergency plans. Differential diagnosis. When whitish, necrotic tail muscles are observed in dead and moribund shrimp, especially in samples from Brazil, Indonesia, India, and Malaysia, IMNV should be included in the list of diseases to be diagnosed. However, this clinical sign can also be caused by a number of factors other than IMNV infection, e.g., hypoxia, overcrowding, abrupt changes in temperature or salinity. IMN is different from the white-tail diseases found in penaeid shrimp or in the giant river shrimp, Macrobrachium rosenbergi, family Pelamonidae, although the clinical and histological signs are similar. These non-IMNV white-tail diseases show macroscopic and histological signs that mimic IMN, but are caused by nodoviruses, Penaeus vannamei nodovirus and Macrobrachium rosenbergi nodovirus. To differentiate between IMNV and PVNV in P. vannamei, and Mr. NV in M. rosenbergi, RT-PCR with virus-specific primers and/or ISH with virus-specific probes or immunodetection assays should be used. Specific molecular tests such as RT-PCR, ISH, or immunodetection tests can be used to differentiate IMNV infection from other non-viral causes. Mortality associated with IMNV is chronic, progressing relatively slowly. However, this type of mortality can also be caused by stress-related bacterial infections. Histological examinations and/or bacterial isolation and enumeration may be used to determine these occurrences and differentiate them from IMNV infection. Control and eradication. Information relevant to the development of management strategies needed to respond to severe outbreaks of IMN or the detection of subclinical IMNV infections in farmed shrimp populations, in particular Penaeus vannamei. The information is more relevant to the shrimp farming industries of Brazil, Indonesia, India, and Malaysia, but the measures described can also be applied to other countries. No effective treatment is available for IMNV-infected shrimp reared on farms. Once established, the disease could persist in wild populations of native shrimp, such as P. subtilis, in Brazil, P. monodon, in Indonesia and India, and P. merguiensis, in Indonesia. Measures to control these wild populations would not be feasible. Control and disposal methods. There are a number of measures that can be taken to prevent the spread of IMNV, including quarantine and movement control, tracing investigation, surveillance, use of LPs without IMNV, use of disease-resistant shrimp, use of probiotics, disinfection of shrimp and eggs/nauplii products, emergency harvesting, destruction of infected shrimp, elimination of sick hosts, decontamination of infected flocks, and vector control. These methods, as well as some environmental considerations and the need for public awareness. Quarantine and travel control. Quarantine and movement restrictions should be implemented immediately if an outbreak of NIDS is suspected. The competent authority shall establish appropriate zone and compartment designations that are management strategies to control, protect the health status of shrimp, and facilitate international trade in shrimp and shrimp products. Zoning relies more on geographical barriers and is usually the responsibility of the competent authority, while compartments are inside production facilities and are managed with biosecurity programs to maintain health status. Detailed information regarding zoning and compartmentalization is available in the Aquatic Code. For zoning, zones can be designated as, see figure. Infected premises or area, the premises, e.g., farm, or area, e.g., geographically separated area, where the infection is present, and its immediate vicinity. This premise is where a presumptive positive case or a confirmed positive case exists based on laboratory results. Buffer zone, an area adjacent to premises or an infected area, which is likely to become an infected area. Free zone, an area that is not infected. A control zone consists of infected premises and a buffer zone. Controls on movement from infected premises, areas, should include prohibition of moving live and uncooked, raw, shrimp from infected premises to VNI-free areas and off-site treatment plants. Restrictions or prohibitions on the discharge of shrimp and pond water from infected premises into aquatic environments. Prohibition of the use of raw shrimp in infected premises as bait for fishing. Prohibition of discharging effluents from processing plants without any treatment into local infected areas. Control of the disposal of dead shrimp. Control of seabird access to live and dead shrimp in infected premises. Restrictions or prohibitions on the use and movement of equipment and vehicles between farms in infected premises. The implementation of these prohibitions will depend on the severity of the disease, the types of operation, such as farm location, farm size, pond system, and the response options chosen. For a fish virus, the infectious salmon anemia virus, a control zone of 5 to 6 kilometers is recommended. Vaccination. There are no effective vaccines against IMNV, because the immune system of shrimp has no memory and is therefore not able to acquire immunity. Immunity and resistance. Shrimp have an innate immune system against viral infections, which involves only nonspecific physiological responses. These include humoral responses involving toll-like receptors, a prophenol oxidase activation system, PROPO, a coagulation cascade and the production of antimicrobial peptides. Cellular immune responses involving apoptosis, encapsulation, phagocytosis, and melanization are also known. The JAK-STAT signaling pathway, which responds to extracellular chemical signals, has also been shown to play a role in the response to viral infections in shrimp. Improvement of disease resistance has been attempted by stimulating the innate immune system, such as responses to prophenol oxidase, by including beta-1,3,1,6 glucan, 0.1%, in shrimp diets. NHPB is a rickettsia-like bacterium that causes necrotizing hepatopancreatitis in several species of penaeid shrimp. NHPB is present in South and Central America and was also detected in Vietnam in 2012. Species mainly cultivated in Latin America such as P. vanamae and P. stylirostris are the most affected. Other susceptible species include Penaeus astecus, Penaeus sativus and Penaeus californiensis. Although NHPB is present in the majority of shrimp, P. vaname, farms in Latin America, its average prevalence of 11.2% remains relatively low. In wild penid species from Latin America, the prevalence varies from 0.5% to 17% depending on the species and the regions of collection. Infections of NHPB on shrimp farms do not necessarily result in infection of nearby wild populations. The following table specifies the imported species, or target species, for necrotizing hepatopancreatitis, depending on whether the imported product comes from aquaculture or wild. Vibrio spp. Species are gram-negative, rod-shaped bacteria found in marine environments and estuarine aircraft from all over the world. Some strains are important pathogens for aquatic species and some can also affect humans. Vibrio spp. Are normally part of the flora of shrimp ponds, but are generally known to be secondary and opportunistic invaders when associated with aquatic animal diseases. Vibrio spp. Are different because they are primary causative agents. They colonize the stomach of shrimp, figure, and secrete the toxin pyrup to the HP, the target organ, causing the epithelial cells to detach and break down, which then became substrates for bacteria to replicate. Eventually, this results in the malfunction and destruction of the HP. Shrimp and prawns affected by upend. VP upend colonized in the stomach of P. vaname. Geographic distribution and prevalence. Limited data are available regarding the persistence of VP upend in the environment. Vibrio perihemolyticus strains show high tolerance to temperature, salinity, and pH due to high metabolic. This allows them to adapt and survive in almost all marine estuarine environments where shrimp farms are located, implying a high risk of disease outbreaks and rapid spread of diseases to disease-free areas. Pathogenic strains of VP upend have been reported to possess properties similar to those of other strains of V. perihemolyticus found in seafood, which have been shown to survive for up to 9 and 18 days in filtered estuarine and seawater, respectively, at an ambient temperature of 28 ± 2 degrees Celsius, and even longer in waters rich in organic matter. Hosts reservoirs. Because Vibrio spp. Are ubiquitous in the marine environment, there are many potential mechanical vectors or environmental reservoirs of VP upend. Vibrio species, including V. perihemolyticus, exist as free-living bacteria, and are often found in association with other aquatic organisms, including phytoplankton, zooplankton, molluscs, crustaceans, polychaetes, and finfish that are common in brackish waters and marine environments. In the ocean, Vibrio spp. Are known to be able to attach to zooplankton, transported over long distances by ocean currents. In shrimp farms, bacteria can be found as biofilms, in pond sediments, or on submerged equipment. In shrimp hatcheries, VP upend are likely to colonize brine shrimp, Artemia spp., which is an essential food used in lavaculture and shrimp maturation. Other potential, but unconfirmed, vectors include crabs, crayfish, and other crustaceans. In addition, VP upend is likely to remain pathogenic in the gut and feces of seabirds that feed on dead or dying shrimp on farms caused by upend. VP upend can be transmitted horizontally via cohabitation or oral ingestion of bacteria. Once the bacteria are defined in a pond, upend can be transmitted rapidly through cannibalism of sick and dead shrimps, or through molts colonized by VP upend. However, the main source of infection was identified as uneaten feed pellets colonized with VP upend. Stopping feeding has been observed to reduce mortality, and while this is a very important observation for understanding disease dynamics, it is not a practical measure to manage the disease. VP upend is present in the digestive tract of infected animals. From oral bioassays, it is clear that individuals can become infected by ingesting sediment and pond water contaminated with the bacteria. There is no evidence of true vertical transmission, i.e., via infected eggs rather than through the surface of contaminated eggs, for VP upand. To prevent possible transmission of VP upand via the egg surface, methods consistent with egg requirements should be applied. A variety of host-specific or environmental risk factors may influence the host's exposure or susceptibility to the pathogen. Environmental conditions that affect both the pathogen and the host shrimp are important determinants of outbreaks. Both the pathogen and the host are affected by conditions such as temperature, salinity, and water quality. Shrimp are more susceptible to disease when stressed by suboptimal rearing conditions. Environmental factors are known to promote upand outbreaks in shrimp farms, including high nutrient concentration, via fertilizers, molasses, etc., high water temperature, high salinity, greater than 5 parts per trillion, high pH, greater than 7, low water exchange, and low planktonic biodiversity and accumulation of organic matter, feed, shrimp carcasses. Factors influencing disease transmission and expression. Some management practices at the pond or farm level may affect the exposure or susceptibility of farmed shrimp to VP upand. These practices can affect shrimp immunity, probiotics, stocking density, pathogen load, disinfection, water changes, environmental conditions, and water quality, feeding, dead shrimp removal, water additives. Farm-level risk factors associated with the onset of upand included having a large growing area, using the sun-dried sediment method to clean the bottom of ponds, and being in close proximity to other farms and using the same water source that is affected by upand. Risk factors at the pond level included water depth of 1.2 meters or less, occurrence of abnormal weather events during the first 35 days of culture, or until the first signs of upand, and the use of fertilizers and probiotics for water treatment. However, this may be due to reverse causation since the use of probiotics can occur in response to a pond with a history of outbreaks. Ponds that were treated with minerals and algicides had a reduced risk of developing upand. Co-infection is common in most cases of upand. Infection with the microsporidian parasite Enterocytozoon hepatopenii, EHP, has been shown to increase the susceptibility of shrimp to upand. Impact of the disease. Mortality in upand shrimp production facilities can be as high as 100% within 35 days, or can have chronic mortality throughout the production cycle. Production losses associated with upand have had a substantial and large-scale impact on shrimp aquaculture. These losses manifest themselves in 1. Production of farmed shrimp, including juveniles in grow-out tanks, broodstock in rearing centers and LPs in hatcheries. 2. Manufacture of animal feed. 3. Processing facilities. 4. International marketing, as buyers are unlikely to purchase live or staple shrimp from countries known to have upand. 5. Employment. Some 2 million people, including temporary and seasonal workers, are involved in shrimp farming and related industries in Asia alone. And 6. Government programs such as compensation for farmers in the event of epidemics and social benefits related to job losses. Additional costs, not yet quantified, would include treatment costs, diagnostic testing, disinfection of facilities, implementation of control measures, inventory tracking, changes in management strategies, and research related to upand. These control programs are often difficult to interrupt due to the risk of new upand incursions. Even if a country is free of upand, there are ongoing costs due to efforts to prevent the introduction of the disease, including surveillance and import controls. The onset of clinical signs and mortality usually begin within 30 to 35 days of restocking in intensive ponds. Mortality rates range from 40 to 100%. Clinical signs include pale to white atrophied HP, figures, an empty stomach and middle intestine, figure, black spots or streaks visible in the HP, due to melanized tubules, and soft shells in the chronic phase of the disease. In addition, HP does not easily crush between the thumb and index finger, likely due to increased fibrous connective tissue and hemocyte accumulation. The macroscopic signs, related to chronic presentation, are not specific to upand. They are also associated with shrimp with necrotizing hepatopancreatitis, NHP, also known as Hepatobacter infection, and other vibrio bacterial diseases. The acute form of this disease can be considered a toxicosis rather than an infection. However, as the disease progresses, there is secondary bacterial colonization on the damaged tissues. Toxins produced by the pathogen Vibrio spp affect the hepatopancreas, HP. Clinical signs include pale to white atrophied HP, figures, empty stomach and middle intestine, soft shell, and occasionally the appearance of black spots or streaks, due to melanization, in the chronically stage HP tubules. However, these clinical signs are not specific and insufficient to make a definitive diagnosis. The occurrence of mass mortality due to UPND in P. Vaname grown in China. Clinical signs of P. Vaname affected by UPND compared to healthy shrimp. Empty stomach and gut, pale atrophied hepatopancreas, principles of control and eradication. Implementing timely and effective actions to contain and eventually eliminate an outbreak of UPND on a farm is important to prevent the spread of the disease. Depending on the disease scenario, one of the following major control approaches, or a combination of these, may be used to respond to an emergency outbreak: I. Eradication. 2. Containment and zoning, or 3. Control and mitigation strategies. The methods described can be used on farms as part of these three main management strategies. Methods of preventing the spread and elimination of pathogens. When an outbreak of UPND occurs, the main objectives to be achieved in response are: eradicate the disease to the extent possible, prevent the spread of disease, and to prevent re-emergence. The specific methods used to achieve these objectives will depend on a variety of factors, including the extent of the outbreak, farm management practices, and marketing issues. These methods, as well as the conditions under which they can be used most effectively, are described in the following subsections. Quarantine and movement control. Quarantine and movement restrictions should be implemented immediately if an UPND outbreak is suspected. Zoning and compartmentalization are management strategies to limit the spread of disease and facilitate international trade in shrimp and shrimp products. Zoning is more based on geographical barriers and is generally the responsibility of the CA, while the compartments are located in the production facilities and are managed by biosecurity programs at the farm level to maintain their health status. Detailed information regarding compartmentalization and zoning is available in the WA Aquatic Code. Zoning is shown schematically in figure and is described in detail below. Designation of zone and facility and the UPND outbreak response. Affected premises, or area, the premises, e.g., a farm, or area, e.g., a geographically separated area, where UPND occurs, and its immediate vicinity. An affected site or area is where a suspected or confirmed positive case exists based on laboratory results in accordance with international standards. Buffer zone, an area adjacent to the affected premises, or zone. Free zone, the unaffected zone. A control zone consists of the affected premises, or zone, and the buffer zone. Controls on movement from the premises, or area, concerned shall include prohibition of moving fresh live shrimp, chilled on ice, from the premises concerned to UPND-free areas. Prohibition of the use of fresh live shrimp in the affected premises as bait for fishing. Prohibitions or restrictions on the release of live shrimp and pond water from affected areas into aquatic environments. Restrictions on the discharge of effluent from the processing plant into the relevant premises. Restrictions on harvesting and transporting shrimp from affected premises to off-site processing plants. Restrictions on the use and movement of equipment and vehicles between farms within affected farms. Control of seabird and crab access to live and moribund/dead shrimp in the affected premises. Control of the elimination of diseased shrimp. The implementation of these prohibitions or restrictions will depend on the severity of the disease. Types of operation, such as farm location, farm size, pond system, and response options chosen. Tracing consists of looking for a. whether the diseased shrimp have been moved to other areas. B. Whether the occupant has spread to other areas, or C. The origin or origins of the disease. It may be necessary to trace the movements of the following items from the affected sites or premises: live shrimp, for example, broodstock, LP and other stocks, including those sold in bait shops. Fresh shrimp, shrimp intended for human consumption or bait. Effluents and wastes from processing plants and farms, discharge into nearby coastal or inland waters. Vehicles, potentially contaminated transport vehicles, food trucks, cars and boats. Agricultural equipment, nets, buckets and other agricultural equipment. Monitoring is continuous systematic sampling of shrimp populations, pond water, sediment and monitoring for the presence of VP upend. It can be used to detect the early onset of upend and determine its prevalence in populations, and is used in the process of maintaining and certifying farms or areas as free of upend. In large shrimp ponds, which can extend over several hectares, monitoring the health of shrimp populations is more difficult. Disease problems, such as mortality or the presence of shrimp with clinical signs, may go unnoticed. Even with rigorous periodic sampling, the probability of detecting disease problems in a large population is low until the problem has progressed significantly. Incubation of pelleted feed in a mesh bag in the suspect pond is recommended. However, careful monitoring of the health of shrimp populations is important to detect problems as early as possible so that strategies, e.g., culling, early harvesting, pond disinfection, and restocking, can be implemented to minimize economic losses. Diagnostic. If an outbreak of upend is suspected, it is important to get an accurate and timely diagnosis. The diagnosis of upend in PL or HP, or stomach, gut, is usually made via a series of procedures after a preliminary diagnosis based on clinical signs. These include histological demonstration of lesions. However, diagnosis by histological methods is generally too slow to allow shrimp farmers to make practical management decisions. Molecular diagnostic methods are much faster and can provide immediate information to shrimp farmers. Molecular detection of VP upend has been successfully developed, with PCR having been widely used for the rapid and specific detection of VP upend. PCR detection kits targeting PYRUB genes are also commercially available. On-site rapid diagnostic tests. There are no reliable vaccines or treatments for shrimp diseases that can be applied to farms, and therefore the shrimp farming industry relies heavily on good farm-level biosecurity programs to prevent the introduction of VP upend into facilities. Optimal on-site detection systems must be fast, inexpensive, sensitive, and easy to maintain and use by non-specialists. In addition, reagents should be supplied in a format that allows for easy shipping and storage. There are a range of different technologies currently available for the rapid diagnosis of upend. LAMP is a simple method involving heating DNA templates and a set of specific VP upend primers to a constant, isothermal, temperature, 65 degrees Celsius, for 45 to 60 minutes. This method is fast, specific and very sensitive. LAMP does not need complex instrumentation, such as a thermocycler as for PCR, nor does it require trained professionals. A commercial VP upend PCR detection test, based on isolated isothermal PCR, using the TACMAN-based qPCR principle, is also available for on-site detection of upend. Destruction and disposal of diseased shrimp. Any chemical agent used to disinfect or destroy shrimp affected by upend must be approved for that use by the NOC. The national or local competent authority should also be consulted for advice before chemicals are used. The slaughter of sick shrimp must be both hygienic and humane, and avoid spills or leaks into the environment. To prevent the mechanical spread of VP upend during the destruction process, scavengers, such as seabirds, should be kept away by the use of electric barriers or nets. Moribund and dead shrimp affected by upend contain VP upend. Therefore, they must be destroyed and disposed of immediately and appropriately, along with other possible infectious waste or sources of bacteria, such as potential aquatic carriers, to reduce the risk of disease. Burying is the cheapest and most convenient method for large quantities of diseased shrimp. The landfill should be located away from shrimp farming areas to reduce the risk of VP up and entering natural waters or susceptible species being exposed to these bacteria. Since heat effectively destroys pathogenic bacteria, incineration can also be used. However, the incinerator must be able to treat the water content involved. See the WA Aquatic Code for more details on disposal methods. Disinfection of affected farms. The disinfection of a farm affected by UPND involves the destruction of stocks and the disinfection of all components of the facility. Many disinfectants are used in shrimp farming to disinfect water before storing shrimp in ponds and disinfect makeup water used to replace evaporation and seepage into ponds. Chlorination is widely used to disinfect municipal water supplies, and a few studies have shown its effectiveness in disinfecting shrimp farm water supplies at 20 to 30 parts per million, ppm. Effective dose rates for disinfectants commonly used in shrimp farming can be established by microbial biological assays. As VP UPND remains viable in pond water, the water should be disinfected by adding chlorine at a concentration of 50 parts per million, with the water being retained for 4 days before being discharged. The sediments in the pond must then be completely dried in the sun for 2 months and can also be treated with a minimum of 6 tons/ha of lime, SO2.
Equipment, e.g., nets, boots, aerators, hoses, and surfaces of tanks, and surrounding facilities, should be thoroughly disinfected, as VP UPND can form biofilms on their surfaces.
Vector control. A wide range of aquatic plants and animals, including phytoplankton and zooplankton, can serve as vectors for VP UPND, allowing the fixed bacteria to be dispersed in shrimp ponds and transported to and through other aquatic ecosystems. These bacteria can colonize and form biofilms on pond sediments and other surfaces, such as those on equipment, pipes, and tanks in aquaculture facilities. In ponds, when these bacteria break off, they disperse and reattach themselves to other substrate-rich surfaces. Agricultural management strategies to reduce the growth of VP UPND include vector control through the use of predatory fish, such as tilapia, disinfection of pond water prior to use, prevention of pond sediment accumulation, and drying of the pond bottom between manufacturing runs. Seabirds are known to be important vectors of shrimp pathogens. They are commonly found around shrimp farms, where they feed on dead and moribund shrimp at the edge of ponds. They often disgorge diseased shrimp in other ponds, spreading the bacteria to unaffected ponds. In addition, scavenging seabirds defecate in the surrounding waters and ponds, contaminating unaffected areas. Therefore, seabird access to shrimp ponds affected by the UPND must be controlled. The installation of nets at breeding sites and the use of deterrents such as polyethylene bags, thermocol and hot crackers are effective in this regard. To the extent possible, contact between crabs and shrimp affected by UPND should also be avoided. Crab intrusion can be prevented by building fences around the rearing sites. Farm management. The implementation of good aquaculture practices to improve biosecurity in shrimp farms is very important to prevent the introduction and spread of VP UPND and other major shrimp pathogens during grow-out. Prior to implementing a site-specific biosecurity action plan, the epidemiology of UPND should be thoroughly investigated to understand the risk factors and pathways, points of entry, associated with the introduction and transmission of VP UPND on a farm. UPND can be introduced through 5 main pathways: affected LPs, water, fomites, vectors, and fresh foods. Control measures can be implemented based on this knowledge to potentially mitigate the risk of introduction and spread of VP UPND in shrimp farms.
To reduce the risk of introducing VP UPND, new animals introduced to a farm must come from a reputable supplier and have a known health status, e.g., specific pathogen-free, SPF, stock, pathogen-free certification. In recirculating aquaculture systems, RAS, or raceway systems, the incoming water must be clean and free of pathogens of concern. This can be achieved by sourcing pathogen-free water, e.g., spring water, well water, or by treating the influent water before it enters the system.
In ponds, VP append can be found as biofilms in sediments or as free-living bacteria. Pond bottoms should be cleaned by disinfection, prior to water drainage, sludge removal, dewatering, and liming prior to restocking to reduce the risk of VP append contamination. Equipment and vehicles can be used as fomites for the transmission of VP append and should therefore be disinfected prior to transfer between sites, ponds or reservoirs. Vectors, although not directly affected by VP append, can serve as transmission vehicles for the bacterium and include species of shellfish, crustaceans, and seabirds. People can also act as vectors by carrying the pathogen on their hands, shoes, and clothing.
Vectors can be controlled with fencing around rearing sites, nets over ponds or tanks, pond disinfection/drying, and predatory fish, e.g., tilapia.
Procedures must be in place for staff and visitors, such as restricting access, sign-in/out, protective clothing, disinfecting shoes and hand-washing stations. The power supply can serve as a source of introduction of VP append and, therefore, should be purchased from a trusted supplier.
Live foods, e.g., Artemia spp, rotifers, can act as vectors of the pathogen and must be certified free of VP append.
The level of VP append in the pond environment will increase or decrease depending on the available organic load. Therefore, overfeeding control is essential to keep VP append at lower numbers. Good and frequent drainage of the pond will facilitate the control of bacterial growth. Stress has an immunosuppressive effect on animals, increasing their susceptibility to infections. Therefore, the use of good farm management practices on farms that limit stress and improve shrimp health are important to reduce the risk of disease spread. This includes maintaining optimal water quality parameters and stocking density, limiting handling, and reducing the presence of predators. Traditionally, antibiotics have been considered as a therapeutic agent in aquaculture farms and hatcheries to control bacterial diseases. Several antibiotics such as oxytetracycline, OTC, tetracycline, quinolones, sulfonamides and trimethoprim are authorized for aquaculture in Asia. The use of antibiotics cannot be blind. Their efficacy should be assessed by susceptibility testing of VP append isolates in laboratories. The use of antibiotics, however, is generally problematic. Use in open systems can lead to contamination of the environment, and use in closed systems can kill beneficial bacteria. In addition, the excessive use of antibiotics generates a high selective pressure that can lead to the transfer of resistance genes associated with plasmids or transposons between bacterial species. Antimicrobial resistance, AMR, is one of the most important issues in public health, veterinary medicine and aquaculture. As a result, the use of several antibiotics important for human medicine is banned or restricted in many countries.
For example, chloramphenicol, fluoroquinolones, e.g., ciprofloxacin and enrofloxacin, and nitrofurans are banned by the government of Vietnam.
There is already evidence of antibiotic-resistant strains of VP append. Due to the problems with antibiotic use, phage therapy has become a promising alternative and natural method for controlling bacterial diseases. Phages, bacteriophages, are viruses that destroy specific bacteria through effective bacteriolytic activity. They are abundant natural inhabitants of all aquatic ecosystems, and therefore it is easier to obtain regulatory approval. Phage therapy uses phages specific to the targeted pathogenic bacteria. Among the advantages of using phages for disease control is the fact that they self-replicate, affect only the targeted bacteria, are relatively inexpensive, and are easily applied in the field, administered through the diet or by direct release into the water. Phage therapy does not cause any of the side effects associated with antibiotic use. Phage therapy has been successful in controlling shrimp diseases caused by the pathogenic Vibrio and has shown potential for use in the control of append. Another approach focused on the use of probiotics. Probiotics are microbial dietary supplements or water additives given to 1. Improve water quality. 2. Improve physiological and immunological responses in aquatic animals, and 3. Reducing the Use of Chemicals and Antibiotics in Aquaculture, HI, 2015. These can be used alone or in combination with prebiotics, non-digestible fiber, such as sweet potato starch, or immunostimulants, such as beta-1,3 glucan. The probiotic agents used successfully in shrimp aquaculture mainly belong to the genera Bacillus, Lactobacillus, Enterococcus, Carnobacterium, Saccharomyces, and Vibrio. Vibrio alginolyticus is a non-pathogenic species used in shrimp and salmon aquaculture. Probiotic supplements are usually provided in food or applied directly to water. The modes of action are mainly via competitive exclusion, the production of substances that inhibit pathogenic organisms, and immunomodulation. However, they can also have antagonistic activity and can improve health, aquatic animal growth, and water quality. Shrimp have an innate immune system that does not have a memory to produce antibodies following exposure to pathogens. This excludes the use of traditional vaccines against specific pathogens to prevent shrimp diseases. The immune system of shrimp, however, responds to harmful microorganisms through complex interactions of cellular and humoral processes. Cellular immune responses, occurring in hemocytes, involve phagocytosis, encapsulation, cellular otoxicity-mediated cytolysis, coagulation, and apoptosis. Humoral responses, which occur in hemolymph, involve the production of proteins, antimicrobial peptides, and other compounds to fight bacteria, fungi, and viruses. From shrimp with upland, comparative transcriptomic analyses have shown that many immune-related genes and signaling pathways are differentially expressed in the stomach, HP, and midget. There are no vaccines against shrimp diseases. However, there are two recent lines of research to obtain tolerance to upland. One is exposure of shrimp to inactivated VP upland or the recombinant PER of protein, and the other is the use of antibodies, extracted from chicken egg yolk, in the diet of shrimp. TAURA syndrome, TSV, is a viral disease of penaeid shrimp caused by infection with the TAURA syndrome virus, TSV. The main host species in which the TSV can cause epidemics and significant mortalities are Penaeus vannamei and P. stylirostris. TSV has three distinct phases: acute, transient, and chronic. In acute disease, pathognomonic lesions are usually present in histological sections of the cuticular epithelium, while in the transitional and chronic phases of the disease, there are no pathognomonic lesions and molecular and antibody-based methods for the detection of TSV are required for diagnosis. Chronic infections in individual shrimp can persist for life. The TSV was listed as an unassigned species of the family Dicystroviridae in the most recent report of the International Committee on Taxonomy of Viruses by Bonamy and Mari. At least 4 genotypes, strains, have been documented, based on the sequence of VP1, the largest and presumably dominant structural protein of the virus. Based on the sequence of VP1 equals CP2, these genotypic groups are 1. The Americas group, 2. The Southeast Asia group, 3. The Belize group, and 4. The Venezuelan group. At least 2 distinct antigenic variants of the TSV have been identified using the monoclonal antibody, MAB1A1, produced against a reference isolate from the Americas. Type A represents those that react to MAB1A1 in the enzyme-linked immunosorbent assay, ELISA. Western blots and in situ hybridization, ISH, with infected tissues, and those that do not. The MAB1A1 non-reactors were subdivided into types P, TSV98 Sinaloa, Mexico, and type C, TSV02 Belize, based on host species and virulence. Sensitive host species. The main host species of the TSV are the Pacific white shrimp, Penaeus vannamei, and the Pacific blue shrimp, P. stylirostris. While the main host species of the TSV all belong to the penaeid subgenus Litopenaeus, other penaeid species can become infected with the TSV by direct challenge, although signs of the disease do not develop. Natural and experimental hosts documented for VST include P. certiferus, P. schmidti. P. monodon, P. chinensis, P. japonicus, P. aztecus, P. duorum and metapaneus. Susceptible host stages. VST has been documented at all life stages, LP, juveniles, and adults, of P. vanameae, the economically most important of the two major host species, except in eggs, zygotes, and larvae. Target organs and infected tissues. TSV infects and has been shown to replicate, using ISH with specific DNA probes, primarily in the cuticular epithelium of the general exoskeleton, foregut, hindgut, gills, and appendages, and, often in connective tissues, hematopoietic tissues, lymphoid organs, oles, and antennal glands. The enteric organs, endoderm-derived hepatopancreas, mucosal epithelium of the checker of the middle and middle intestine, and smooth, cardiac, striated muscle, as well as the ventral nerve cord, its branches, and lymph nodes usually show no histological evidence of TSV infection and are usually negative for TSV by ISH. Persistent infection with lifelong carriers. Some members of P. vanameae or P. stylirostris populations who survive TSV infections and/or epizootics may carry the virus for life and, although undocumented, transmit the virus to their offspring through vertical transmission. Survivors of TSV infections can carry the virus for life. Seabirds, TSV has been shown to remain infectious for up to 48 hours after ingestion of VST-infected shrimp carcasses in the feces of wild or captive gulls, Larus drusilla, and chickens, Gallus domesticus, used as a laboratory surrogate for all shrimp-eating birds. These findings implicate birds as an important mechanical vector for transmission of the virus within affected farms or agricultural regions. Aquatic insects, the aquatic boatman, Trichocoryxa reticulata, Coryxidae, an aquatic insect that feeds on shrimp carcasses in shrimp ponds, has also been shown to serve as a mechanical vector of TSV. Frozen commodities infected with TSV, found in frozen shrimp and prawn, P. vanameae, commodities in samples from markets in the United States from Latin America and Southeast Asia. Improper disposal of waste, liquids and solids, shells, heads, intestinal tracts. Value-added reprocessing of SWT-infected shrimp at inshore sites can provide a source of SWT that has the potential to contaminate wild or farmed stocks near the point of discharge of the waste stream. Zoonotic potential. TSV has been reported to infect human and monkey cell lines, suggesting zoonotic potential for this virus and that penaeid shrimp may serve as reservoirs for TSV and other members of the picornavirus superfamily that infect humans. Mechanisms of transmission. Transmission of TSV can occur through horizontal or vertical routes. Horizontal transmission by cannibalism or contaminated water has been demonstrated. Vertical transmission from infected adult broodstock to their offspring is strongly suspected but has not been confirmed experimentally. Prevalence. In areas where the virus is enzootic in farms, the prevalence of TSV has been found in various surveys ranging from 0 to 100%. Geographical distribution. TSV is widely distributed in the shrimp farming regions of the Americas and Southeast Asia. Americas, following its recognition in 1992 as a separate disease of P. vanameae farmed in Ecuador, TS has spread rapidly in many shrimp farming regions of the Americas through shipments of PL and infected broodstock. In the Western Hemisphere, TS and TSV have been reported in virtually all penaeid shrimp farming regions of the Americas and Hawaii. TSV is enzootic in farmed penaeid shrimp stocks on the Pacific coast of the Americas from Peru to Mexico, and has occasionally been found in some wild P. vanameae stocks in the same region. TSV has also been reported from cultured penaeid stocks on the Atlantic, Caribbean and Gulf of Mexico coasts of the Americas, but has not been reported from wild stocks in these regions. Asia. TSV was introduced to Chinese Taipei in 1999 with infected imported Pacific white shrimp, P. vanameae, from Central and South American sources. Since this initial introduction, The virus has spread with movements of broodstock and LP to China, People's Republic of Thailand, Malaysia and Indonesia, where it has been the cause of major epizootics with high mortality rates in unselected introduced stocks of P. vannamei. Mortality and morbidity. In on-farm epizootics of TS involving unselected stocks of P. vannamei, the main host species of TSV, typical cumulative mortalities range from 40 to greater than 90% in cultivated populations at the PL, juvenile and subadult stages. TSV-resistant P. vannamei lines are available and have survival rates of up to 100% in laboratory testing with all 4 TSV genotypes. Environmental factors. Outbreaks of TSV are more frequent when salinities are below 30 parts per trillion. No effective vaccine against TSV is available. Resistance breeding. Pannaea stylirostris was found to be resistant to TSV, genotype 1, MAB1A1 type A, after the emergence of TS in Ecuador in 1992 to 1994. Restocking with TS-resistant P. vannamei species have been developed and are commercially available. Blocking agents. Resistance to TSV infection was signaled by the expression of the antisense RNA of the TSV envelope protein in P. vannamei zygotes. Transgenic juveniles reared from zygotes protected in this way showed improved resistance to challenge by TSV by oral or intramuscular (IM) injection. Similar results were produced by introducing random double-stranded RNA sequences into juvenile P. vannamei. Disinfection of eggs and larvae. Although TSV is thought to be transmitted vertically, transovarian transmission, no reports documenting this route of transmission have been published. Disinfection of eggs and larvae is a good management practice and is recommended for its potential to reduce contamination of laid eggs and larvae. General breeding practices. Some husbandry practices have been successfully applied to the prevention of SVT infections and TS disease. These include the application of the reaction preselection chain polymerase, PCR, of wild or pond-reared broodstock and or their eggs/nauplius laid and the removal of those with positive virus tests, the fallowing and restocking of entire growing regions with SVT-free stocks, and the development of shrimp stocks free of specific pathogens, SPFs, of P. vannamei and P. stylirostris, selection of individual specimens. Suitable specimens for testing for TSV infection include LPs, juveniles, and adults. Although the virus can infect all life stages, the severity of infection, and therefore the viral load, may be below the limits of detection in laid eggs and larval stages, so these life stages may not be appropriate samples for the detection of VST or certification of the absence of SW disease. Pooling of samples. Samples collected for molecular testing may be combined as pooled samples of up to 5 specimens per pooled sample of fecal filaments, juveniles, subadults and adults. However, for eggs, larvae, and aggregation of LPs, larger numbers, about 150 or more eggs or larvae or 50 to 150 LPs depending on their size/age, may be required to obtain sufficient sample material, extracted nucleic acid, to perform a diagnostic test. Better organs or tissues. TSV infects tissues of ectodermal and mesodermal origin. The main target tissue in the acute phase of ST is the cuticular epithelium. In chronic infections, low is the primary tissue target. Excised hemolymph or pleopods may be collected and used when non-lethal testing of valuable broodstock is required. Samples/fabrics that are not suitable.
TSV is a systemic virus and does not replicate in enteric tissues, e.g., hepatopancreas, midget, or its checker.
Therefore, enteric tissues are inappropriate specimens for the detection of TSV infection. Only the acute phase of TS can be presumptively diagnosed from clinical signs. Behavioral changes. Only shrimp with the acute phase of TS disease show behavioral changes. Typically, severely affected shrimp become hypoxic and move to the edges or surface of the pond where dissolved oxygen levels are higher. These shrimp can attract seabirds in large numbers. In many outbreaks of TS, it is the large number of seabirds attracted to moribund shrimp that first indicates the presence of a serious outbreak, which is often either TS or WSD when seabirds are observed, to the farm manager. Macroscopic pathology. TSV disease has three distinct phases: acute, transitional, and chronic, which are roughly distinguishable. The crude signs exhibited by juvenile, subadult, and adult shrimp in the transitional phase of TSV are unique and provide a tentative diagnosis of the disease. Acute phase, coarse signs displayed by moribund P. Vaname with acute phase TS include expansion of red chromatophores, giving the affected shrimp a pale reddish general coloration and making the caudal fan and pleopods distinctly red. Therefore, the red tail disease was one of the names given by farmers when the disease first appeared in Ecuador. In these shrimp, careful inspection of the cuticular epithelium in thin appendages, such as the edges of uropods or pleopods, with a ×10 magnifying glass reveals signs of focal epithelial necrosis. Shrimp showing these coarse signs of acute TS usually have shells, an empty intestine, and are often at the end of the D stages of the molting cycle. Severely affected shrimp usually die during molting. If the affected shrimp weigh more than about 1 gram, the moribund shrimp may be visible to seabirds at the edges and surface of the pond. Thus, during the peak of severe epizootics, hundreds of seabirds, gulls, terns, herons, cormorants, etc., can be observed feeding on infected moribund shrimp that accumulate on the surface and edges of the affected pond. Transition phase, recovery. Although present for only a few days during TS epizootics, the crude signs presented by shrimp in the transition phase can provide a provisional diagnosis of TSV infection. During the transition phase, which can occur when many shrimp in affected populations are still in the acute phase and daily mortality is high, a medium to moderate number of shrimp in affected ponds have random, multifocal, irregularly shaped melanized cuticular lesions. These melanized spots are hemocyte accumulations indicating the sites resolving the TSV lesions in the epicutic thelium. These shrimp may or may not have soft cuticles and red chromatophore expansion, and may behave and feed normally. Chronic phase. After successfully molting, shrimp in the transition phase enter the chronic phase of TSV, in which persistently infected shrimp show no obvious signs of disease.
However, P. vaname chronically infected with TSV may be less resistant to normal environmental stresses, reductions in salinity, than uninfected shrimp.
TSV disease in the acute and chronic phases can be diagnosed most reliably using histological methods. The pathognomonic pathology induced by TSV is unique in acute phase infections. In chronic TSV infections, the only lesion usually exhibited by infected shrimp is the presence of an enlarged low with several low spheroids, LOS, which are indistinguishable from loss induced by chronic infections of other RNA viruses. When loss is observed by routine histology and chronic TSV infection is suspected, molecular testing, ISH with TSV-specific probes or reverse transcription, RT-PCR, is recommended to confirm TSV infection. Acute phase. The diagnosis of TSV in the acute phase of the disease depends on histological evidence, in hematoxylin and eosin, Handa-stained preparations, of multifocal areas of necrosis in the cuticular epithelium of the general body surface, appendages, gills, hindgut and foregut, the esophagus, the anterior and posterior chambers of the stomach. The cells of the subcutaneous connective tissues and the striated muscle fibers adjacent basal to the affected cuticular epithelium are sometimes affected. In some severe cases of acute TS, the epithelium of the antennal gland tubule is also destroyed. Prominent in multifocal cuticular lesions are visible foci of affected cells that exhibit increased eosinophilia of the cytoplasm and pycnotic or charetic nuclei. These structures, together with the pycnotic and charetic nuclei, give the acute phase TSV lesions a characteristic peppery or buckshot appearance, which is considered pathognomonic of ST disease when there is no concomitant necrosis of the low tubule parenchymal cells, The absence of low necrosis in acute phase TSV infections distinguishes TS disease from yellowhead disease in acute phase, in which necrosis patterns similar to those induced by TSV may occur in the cuticular epithelium and gills. In TSV infected tissues, pycnotic or charetic nuclei give a positive Fulgen reaction for DNA, which distinguishes them from less basophilic to eosinophilic cytoplasmic inclusions that do not contain DNA. The absence of hemocyte infiltration or other evidence of a significant inflammatory response from the host response distinguishes the acute phase of TS from the transient phase of the disease. In the transition phase of TS, cuticular lesions typical of the acute phase decrease in abundance and severity and are replaced by visible infiltration and accumulation of hemocytes at the sites of necrosis. Masses of hemocytes can melanize by giving rise to the irregular black spots that characterize the transitional phase of the disease. In hand dissections, such lesions may show cuticle erosion, surface colonization, and invasion of the affected cuticle and surface hemocytes exposed by Vibrio spp. Chronic phase of TAURA syndrome. Shrimp in the chronic phase of TS show no obvious signs of infection and, histologically, the only sign of infection is the presence of many prominent losses, which may remain associated with the main body of the paired lobe, or which may break off and become ectopic loss.
Bodies that are lodged in the constricted areas of the hemocell, i.e., the heart, gills, in the subcutaneous connective tissues, etc.
Such loss are spherical accumulations of low cells and hemocytes and can be distinguished from normal low tissues by their spherical nature and the absence of the central vessel that is typical of normal low tubules. When tested by ISH with a cDNA probe for TSV, or with MAB1A1 using ISH, some cells in the loss give positive reactions to the virus, while no other target tissue responds. Direct microscopy on wet media. Direct microscopy of simple wet unstained assemblies from excised pieces of gills, appendage tips, examined by phase or reduced light microscopy can be used to demonstrate and make a tentative diagnosis of acute phase TS, focal lesions of acute phase TS in cuticular epithelial cells. Preparations with acute phase lesions of TS will contain numerous spherical structures, see histopathological methods, which are pycnotic and charetic nuclei and cytoplasmic remnants of necrotic cells. The currently available methods for targeted surveillance and diagnosis of VOF are listed in the table. The designations used in the table indicate A equals the method is the recommended method for reasons of diagnostic availability, utility, specificity and sensitivity. Recommended tests for targeted monitoring to report the absence of TAURA syndrome. As shown in the table, RT-PCR is the recommended method for targeted monitoring of reasons for diagnostic availability, utility, specificity, and sensitivity. In investigations of acute mortality episodes as part of a targeted surveillance program, demonstration of TSV-induced pathognomonic lesions in the cuticular epithelium by histology, with or without ISH confirmation with TSV-specific DNA probes, is an appropriate method. Table. A suspected case is represented by sudden and high mortalities in late LPs, juvenile or subadult P. Vaname or P. stylirostris in regions where the TSV is enzootic. The sudden presence of many seabirds, gulls, cormorants, herons, terns, etc., fishing in one or more shrimp farming basins. Samples of P. Vaname or P.
stylirostris reared from seabird feeding ponds that show clear signs of an acute or transitional phase of TS, such as general reddish discoloration, lethargy, soft shells, empty intestines and numerous irregular black spots on the cuticle, or demonstration of foci of necrosis in the cuticular epithelium using low magnification, i.e., a magnifying glass times 10.
Or by direct microscopic examination of wet mounts, to examine the edges of appendages such as uropods or pleopods, or gills. Definition of confirmed case. Any combination of a molecular test, PCR or ISH, and a morphological test, histology, using two or more of the following three methods, with positive results. Histological demonstration of acute phase diagnostic lesions of TSV in particular, The cuticular epithelium of the foregut, esophagus, anterior or posterior chambers of the stomach, and/or in the gills, appendages or general cuticle.
Such TSV lesions are pathognomonic to TSV only when they occur without accompaniment of severe acute necrosis, with nuclear pycnosis and chirexis, of parenchymal cells of lymphoid organ tubules, which may occur in acute yellowhead infections. ISH-positive signal, with a TSV-specific cDNA probe, to TSV-like lesions in histological sections, i.e., cuticular lesions of acute phase TS, or to distinctive lymphoid organ spheroids, LOS, in the lymphoid organs of shrimp with chronically phased TS lesions. Positive RT-PCR results for TSV. Sequencing of the PCR product encompassing CP2 can be performed, if necessary, to determine the genotype of the TSV.
The white spot syndrome, WSSV, virus was first reported in shrimp aquaculture in Taiwan in the early 1990s. Since then, the virus has spread around the world and has had a major negative impact on shrimp aquaculture. The virus was recently designated as the only member of a new family of viruses, the Nemaviridae. Nemaviridae are a newly recognized family. The concept of species will be subject to change after existing and new isolates have been studied in more detail. Etiological agent strains WSSV has been designated by the International Committee on Taxonomy of Viruses, ICTV, as the only member of the genus Whispervirus within the family Nemaviridae. Today, although various geographic isolates with genotypic variability have been identified, they are all classified as a single species, white spot syndrome 1 virus, within the genus Whispervirus. Survival outside the host. The agent is viable for at least 30 days at 30 degrees Celsius in seawater under laboratory conditions, and is viable in ponds for at least 3 to 4 days. Agent stability. Effective inactivation methods. The agent is inactivated in less than 120 minutes at 50 degrees Celsius and less than 1 minute at 60 degrees Celsius. Life cycle. In vitro studies on primary cell culture and in vivo studies on post-larvae, PL, show that the replication cycle is about 20 hours at 25 degrees Celsius. Reception factors. WSSV has an extremely wide host range. The virus can infect a wide range of aquatic crustaceans, including marine, brackish and freshwater penaeids, crabs and crayfish. Susceptible host species. All decapod crustaceans, order Decapoda, from marine, brackish or freshwater sources are sensitive host species. Susceptible host stages. All life stages are potentially susceptible, from eggs to spawners. Species or subpopulation predilection, probability of detection. There is a higher probability of detecting the virus in crabs than in shrimp. The best life stages of crustaceans for detection are the late PL stages, juveniles, and adults. The probability of detection may be increased by exposure to stressful conditions, e.g., removal of the eye rod, spawning, molting, changes in salinity, temperature, or pH, and during plankton blooms. Target organs and infected tissues. The main targets of WSSV infection are ectodermal and mesodermal embryonic tissues, particularly the cuticular epithelium and subcutaneous connective tissues. Although WSSV infects the underlying connective tissue in the hepatopancreas and midget of shrimp, the hepatopancreatic tubular epithelial cells of these two organs are of endodermal origin and do not become infected. Persistent infection with lifelong carriers. Persistent infection is common and lifelong infection has been demonstrated. Viral loads during a persistent infection can be extremely low and potentially undetectable by any available diagnostic test. Vectors. Vectors include rotifers, marine molluscs, polychaete worms and non-decapod crustaceans including Artemia salina and copepods, as well as non-crustacean aquatic arthropods such as sea slaters, Isopoda, and insect larvae Euphydradae. All of these species can accumulate high concentrations of viable WSSV, although there is no evidence of viral replication. No suspected carriers of wild aquatic animals have been reported. Infection sometimes causes disease and sometimes not, depending on factors that are still poorly understood but are related to species tolerance and environmental triggers. With an appropriate dose of infection to allow sufficient time before mortality, disease-susceptible animals have a large number of virions circulating in the hemolymph, but this can also occur for tolerant species that show no mortality. Thus, high viral loads in and of themselves do not cause disease or mortality for all susceptible species. Mechanisms of transmission.
Infection can be transmitted vertically, transovular, horizontally through consumption of infected tissues, e.g., cannibalism, predation, and through water.
Transmission of the infection can occur from apparently healthy animals in the absence of disease. Dead and dying animals can be a source of disease transmission. Prevalence. The prevalence varies widely, from less than 1% in infected wild populations to 100% in captive populations. Geographical distribution. WSD has been identified in crustaceans in China, Japan, Korea, Southeast Asia, South Asia, the Indian continent, the Mediterranean, the Middle East, and the Americas. Free WSD zones and buckets are known in these regions. Mortality and morbidity. All species of pennied shrimp are highly susceptible to infection, often resulting in high mortality. Crabs, crayfish, freshwater shrimp, spiny lobsters and clawed lobsters are susceptible to infection, but morbidity and mortality resulting from infection are highly variable. High-level infections are known in some decapods in the absence of clinical disease. Environmental factors. Outbreaks can be induced by stressors, such as rapid changes in salinity. Water temperature has a profound effect on disease expression, with average water temperatures below 30 degrees Celsius being conducive to WSD outbreaks. Control and prevention. Although the underlying mechanism remains unknown, laboratory experiments have shown that vaccinated shrimp and crayfish have better survival rates after a challenge to WSSV. It was first shown that Panas japonicus shrimp that survived natural and experimental WSSV infections showed resistance to further WSSV provocation. Subsequent studies showed that intramuscular injection of inactivated WSSV virions or recombinant structural protein, VP28, offered shrimp some protection against experimental WSSV infection. Immunostimulation. Several reports have shown that beta-glucan, vitamin C, algae extracts, fucoidan, and other immunostimulants improve resistance to WSD. Disinfection of eggs and larvae. For transovular transmission, egg disinfection is likely to be effective, but this has not yet been confirmed by formal scientific trials. General husbandry practices. A number of breeding practices have been used successfully to manage WSD, such as avoiding storage during the cold season, using seed stocks that are free of specific pathogens, SPFs, or negative for polymerase chain reaction, PCR, and the use of biosecure water and cultivation systems. Sampling. Selection of individual specimens. Samples of moribund shrimp or shrimp that exhibit clinical signs or behavioral changes should be selected for WSSV detection. Better organs or tissues.
Analysis of tissue tropism of experimentally infected shrimp and wild-caught female farmers shows that tissues from the ectoderm and mesoderm, especially the cuticular epithelium and subcutaneous connective tissues, as well as other target tissues, e.g., antennal gland, hematopoietic organ, are the main target tissues for WSSV.
It is recommended that samples be submitted from the pleopods, gills, hemolymph, stomach, or abdominal muscles. For non-destructive PCR screening, it is recommended to submit a small piece of gill, a small aliquot of hemolymph, or a small piece of pleopod. There is also evidence to suggest that an ablated eye stem would be a good alternative, provided that the compound eye is removed prior to submission. Clinical signs. White spots embedded in the exoskeleton are the most common clinical sign observed. In most shrimp, these spots range from barely visible to 3 mm in diameter, and sometimes coalesce into larger patches. However, It should be noted that environmental stressors, such as high alkalinity or bacterial diseases, can also cause white spots on shrimp's shells, and moribund shrimp with WSD may actually have few or no white spots. Therefore, the appearance of white spots is not a good diagnostic sign of a WSSV infection. In addition, other crustaceans, such as most crayfish, often show no signs of white spots when infected with WSSV. High degrees of color variation with a predominance of reddish or pinkish discolored shrimp are observed in diseased populations. Behavioral changes. The presence of white spots does not always mean that the condition is terminal. For example, under non-stressful conditions, infected shrimp that have white spots can survive indefinitely. However, if the shrimp also appear lethargic, if their color turns to pink or reddish-brown, if they congregate on the edges of ponds/reservoirs at the surface of the water, or if there is a rapid reduction in food consumption, a very high mortality rate in the shrimp population can be expected within a few hours to a few days of the onset of these signs. The currently available methods for targeted surveillance and diagnosis of WSSV are listed in the table below. Recommended tests for targeted surveillance to declare the absence of white spot disease. Two-step PCR and sequencing are the recommended methods for reporting absence, only for juveniles and adults and possibly LPs. Negative two-step PCR results are required. When a positive two-step PCR result cannot be confirmed by sequencing, this also counts as a negative result. Corroborating diagnostic criteria. Definition of suspected case. For juvenile and adult shrimp, clinical signs of WSD. For shrimp at any stage of their life, from larva to adult, mortality. For shrimps and crabs at all life stages, from larva to adult, enlarged nuclei in gill and or cuticular epithelial squash preparations, unusual aggregates in hemolymph by dark field microscopy, inclusion body in histological sections of target tissues. Confirmed case definition. Suspected cases must first be checked by PCR. If in a country/area/compartment previously free of WSSV, PCR results are positive, they should be confirmed by sequencing. Histopathology, probes, and electron microscopy may also be used to confirm the case. Whitish muscle disease of Macrobrachium rosenbergi, or whitish disease, WTD, is a disease of the giant freshwater shrimp Macrobrachium rosenbergi caused by a nodovirus that has not yet been officially classified. It generates considerable economic losses in the Caribbean and in Asia, reported for the first time in the French West Indies, then in China, India, Chinese Taipei, and Thailand. Prevalence ranges from 10% to 100% in hatchery, nursery and grow-out systems, as well as in experimental immersion challenge, and 100% mortality has been reported 5 to 7 days after the onset of first macroscopic signs of LP in natural or experimental infection. The disease affects the West Indies, the Dominican Republic and the Caribbean, as well as Asia, China, Taiwan, India, and it expresses itself in the same way everywhere. It generates enormous economic losses in commercial giant shrimp farms. Mr. NV has been identified as the causative agent of the disease, and another virus, XSV, for extra small virus, could also be involved. Both are RNA viruses that are routinely found on sick shrimp. The second could be a satellite virus, the first acting as an enabler. The Mr. NV virus, which thrives in both cool and brackish waters, attacks the larval and post-larval stages as well as juveniles. Adults do not seem to be affected, but can serve as healthy carriers. The etiologic agents are two viral pathogens, namely Macrobrachium rosenbergi, nodovirus, Mr. NV, primary, and extra small virus, XSV, related. Mr. NV is important in WTD outbreaks in shrimp, although the role of XSV in pathogenicity remains unclear. The strains are not yet known. Mr. NV belongs to the family Nodaviridae. XSV is the first satellite virus in animals, and it is also the first record of a satellite nodovirus association. Survival outside the host is not known, However, viral inoculum prepared from tissue homogenates stored at -20 degrees Celsius caused 100% mortality in the LP of M. Rosenbergi by immersion challenge. Agent stability, effective inactivation methods. The stability of the agent is not known. However, heat treatment destroyed the infectivity of Mr. NV and XSV in the challenge experiments. Transmission of the viruses can be horizontal or vertical. Infected breeders behave as healthy carriers and generate infected post-larval individuals. Susceptible host species: the giant freshwater shrimp, Macrobrachium rosenbergi. Other known or suspected hosts are not yet known. Larvae, LPs and juveniles are susceptible, while adults are resistant and act as carriers. Target organs and infected tissues: Mr. NV and XSV are confined to the gill tissue, head muscle, heart, abdominal muscle, ovaries, pleopoda, and tail muscle, but not to the hepatopancreas or ocular peduncle. The presence of virus in the ovary tissue indicates the possibility of vertical transmission of WTD from the reproductive stock to the larvae and LP. Experiments have proven that pleopods would be a convenient source of RNA for non-destructive screening of Mr. NV and XSV without stress for shrimp. The disease occurs in the post-larval stage within 3 to 5 days of desalination. Mortality can then reach 30 to 100%. Bacteriological examination shows the presence of Staphylococcus spp, but administration of these microorganisms to healthy shrimp populations does not reproduce the disease. The affected populations are lethargic and anorexic. The abdominal muscles are opaque and the tails whitish. The condition spreads forward and backward, sometimes with degeneration of the telsin and uropods. The size of the cephalothorax increases to double, branchiostachite blister disease or swollen head syndrome. When opened, the cephalothorax sometimes reveals 2 saccular structures swollen with fluid surrounding the hepatopancreas. The histological examination revealed a necrotic musculature. Control. The control of the disease involves screening, observation of post-larval subjects with milky abdomens, mortality, histology compatible with the diagnosis. The definitive diagnosis is obtained by PCR and the ELISA method. There is no cure for the disease, and prevention is the only way to keep the disease out of shrimp farms. Breeding animals and individuals in the post-larval phase must be screened regularly. Infected populations must be eliminated in compliance with health regulations. Farming methods and conditions must be improved. A strict quarantine must accompany any movement of the population, in order to avoid the spread of the disease. General husbandry practices. Experimental infection confirmed the possibility of horizontal and vertical transmission of WTD in cropping systems. Good husbandry practices, such as proper disinfection of tanks, water, and broodstock, and the use of RT-PCR negative broodstock in hatchery grow-out tanks can be helpful in preventing WTD in rearing systems. There is no evidence of prevention of WTD by crop rotation with rice or polyculture with fish. Abalone herpesvirus, ABHV, abalone viral lymph node, GVO. Abalone herpesvirus, ABVH, is the etiological agent of abalone viral ganglion neuritis, GVO, a contagious disease, of abalone species in Australia. Geographical distribution: Australia, Victoria, and possibly Tasmania. Host species: Haliotis levigata, Haliotis rubra and their hybrids, as well as Haliotis cornucopera in Australia, and possibly Haliotis diversicolor supertexta in Taiwan, where cohabiting Haliotis discus have remained normal. Host impact: Outbreaks of GVO in wild and farmed abalone populations in Victoria, Australia, were associated with rapid onset and high mortality rates, up to 90%, in all year classes. Transmission studies have shown that ABHV spreads through direct contact and water, with infectivity easily reduced by dilution. The ABHV was likely carried by the officers on which it was traveling several kilometers. A variable proportion of affected abalone may display an irregular peripheral concave elevation of the foot, turgid, swollen mouthparts that protruded before the podorsal muscle. In severe cases, the radula came out of the mouth prolapsed and the contours of the two odontophores, cartilaginous plaques normally inside the mouth, were visible. In some affected abalone, the radula was completely everted and hanging down. Obvious signs of pustules and vesicles observed on the foot of infected animals are also considered indicative of abalone viral ganglion neuritis. In the field, active signs of viral infection include loss of muscle control in infected animals, minimal movement of the pediosus muscle, excessive mucus production, absence of the marked extension of the foot displayed in the writing reflex when a healthy abalone is turned on its back and the adhesion of its foot to the substrate is reduced. In areas of high mortality, large numbers of intact moribund abalone, shiny empty shells, and flesh in water are characteristic signs of the disease. Paralysis, muscle and mantle stiffness, excessive mucus production and no high mortality were observed in ponds in H. Levigata and H. rubra from the coast of Tasmania, but no high mortality. In Taiwan, clinical signs of disease in H. diversicolor supertexta were mantle recession and muscle stiffness, as well as high mortalities. The main histopathological lesion is ganglion neuritis, proliferation or infiltration of hemocytes confined into nerve tissue from gray matter and extending into white matter and perineural sheath, and possibly associated cell necrosis. Nerve tissues, which include the cerebral, pleurophagia, and oral ganglia, are the best tissues for examining lesions. In addition, lesions were not consistently detected in non-nervous tissues. The lymph nodes and the neurolemma around them can be affected, as can the cerebral commissure and peripheral nerves in some cases. Occasionally, neuron nuclei exhibited notched chromatin with morphological features resembling intranuclear viral inclusions, but no Cowdery inclusion, or herpetic inclusion, was observed. In Taiwan, the nervous system of infected abalone has also been reported to be the primary tissue target. Infected cells, hemocytes and possibly glial cells, sometimes show damage to the nuclear membrane, swelling of the mitochondria and proliferation of the endoplasmic reticulum. The virus replicates in the nucleus, and maturation takes place in the cytoplasm of infected cells. Diagnosis by DNA probes. To detect AB-HV infection in the absence of clinical disease, the AB-HV genome was sequenced and sensitive molecular diagnostic techniques were developed and validated. Control methods. Infected abalone should not be transported to areas that are known to be disease-free. For example, in Western Australia, Jones and Fletcher recommended strategies to mitigate the risk of introducing average into wild abalone stocks through the release of juvenile abalone from hatcheries. The WA recommends the implementation of high levels of biosecurity on the farm and in regional retention and movement facilities. On Australian abalone farms, there was a strong suspicion that stressors, such as high densities, spawning timing, and water temperature, were factors that contributed to the high mortality rates. Tests to evaluate the efficacy of chemical treatments showed that iodophores, such as bufadin at 50 parts per million, and a non-ionic surfactant, such as the detergent Impress at 1%, inactivated the virus after exposure of 10 minutes, while calcium hypochlorite, free chlorine at 20 parts per million, produced only partial inactivation, assessed by injection into novice abalone, cultured hybrids of H. rubra x H. levigata, after exposure to AB-HV to any of the three elements. Storage of AB-HV in liquid nitrogen maintained viral infectivity and pathogenicity for at least 21 months, while isolates stored at -80 degrees Celsius partially lost viability within 12 months and those stored at -20 degrees Celsius lost much of their viability over the same period. A country, zone or compartment declared free of infection with abalone herpesvirus in accordance with the provisions may maintain its disease-free status with regard to abalone herpesvirus, provided that basic biosafety conditions are maintained at all times. A country, zone or compartment declared free of abalone herpesvirus infection may discontinue targeted surveillance while maintaining its abalone virus herpesvirus-free status, provided that conditions conducive to clinical manifestations of abalone herpesvirus infection, as described in the relevant chapter of the Aquatic Manual, are met and basic biosafety conditions are maintained at all times. However, in areas or compartments declared free of infection with abalone herpesvirus in countries infected with abalone herpesvirus, as well as in all cases where conditions are not conducive to its clinical manifestations, targeted surveillance shall be continued at a level defined by the Aquatic Animal Health Service in relation to the probability of introduction of the infection. Intracytoplasmic infections by XENOHALIOTIS californiensis, a rickettsia, in the gastrointestinal epithelium cause a disease, called dieback syndrome, in wild and farmed abalone, Haliotis spp. Crude signs of the disease include pedic atrophy, mottled digestive gland, anorexia, weakness, and lethargy. Common and generally accepted names of the organism or pathogen: foot atrophy syndrome, wasting disease, foot wasting syndrome, abalone wasting syndrome, and intracellular rickettsial prokaryote, WSRLP, wasting syndrome caused by infection with rickettsia-like organisms. XENOHALIOTIS californiensis is an intracellular bacterium of the Anaplasmataceae family, and is closely related to members of the genera Ehrlichia, Anaplasma, and Cowdria. The disease caused by this bacterium is known as wasting syndrome, and may be more accurately referred to as abalone rickettsiosis. Although X. californiensis is considered an obligate intracellular organism, the bacterium can survive outside the host for an indeterminate period of time, as evidenced by waterborne studies. Agent stability. Based on successful laboratory decontamination, this bacterium is easily inactivated by immersion in less than 10% bleach. In addition, exposure of seawater containing the bacterium to greater than 10 mg/liter calcium hypochlorite, and disinfection of the material in a 1% tamed iodine bath in fresh water for 1 hour, are effective disinfectants based on the use of these disinfection methods within a continuous flow marine laboratory seawater, and lack of detection of this pathogen in adjacent abalone populations. Life cycle. The bacterium divides by binary fission, and has direct horizontal transmission. XENOHALIOTIS californiensis infects members of the genus Haliotis, and natural infections have been observed in Atlantic abalone, white abalone, socky abalone, pink abalone, greenslam abalone, small abalone, European abalone, in the wild or in rearing facilities, as well as bull abalone, and Japanese abalone. Temperature is important both in the transmission of pathogens and in the expression of disease. Susceptible stages of the host. While all post-larval stages have been shown to be susceptible to X. californiensis infection, the disease is generally observed in animals over 1 year of age in farmed abalone and all abalone size classes observed in wild populations studied. Species or subpopulation preference. The probability of detection increases with increasing abalone size. Animals less than 10 mm in size have a reduced probability of detection by histology, but an equal probability of detection by PCR. Target organs and infected tissues. The gastrointestinal epithelial cells of the posterior esophagus, digestive gland and, in the intestine. Persistent infection with lifelong carriers. Infections can persist for long periods without developing clinical disease when the host is kept at cool water temperatures, 15 degrees Celsius for swallow abalone, and exposure to high seawater temperatures, greater than 17 degrees Celsius for several, black, and white abalone, usually results in clinical disease.
Variable seawater temperatures with a lower average temperature, e.g., 16.5 degrees Celsius for slippery abalone, can exacerbate losses.
Recent evidence suggests that some species, particularly those living in warmer waters, can harbor the bacterium without developing clinical disease. Vectors. Although no alternative non-halotidal host has been identified, it has been suggested that some colonial sea squirts may concentrate the bacterium, based on PCR evidence. Thus, the possibility that such species could act as vectors for the bacterium exists, but further investigation of possible vectors is warranted. Mechanisms of transmission. The transmission of X. californiensis is horizontal and assumed to be fecal-oral. Exposure of abalone to seawater containing infectious material is sufficient for transmission of the bacterium, and no direct contact with animals is required. Temperatures below 13 degrees Celsius have been shown to limit transmission of the bacteria, less 1% transmission, compared to those maintained at about 18 degrees Celsius, 72 to 94% transmission. Prevalence. A. Prevalences of 1 to 17% have been observed in Northern California and up to 75% in Central and Southern California. B. Great abalone generally have a higher prevalence of infection. C. Only 36 animals were sampled from a farm in Thailand. Geographical distribution. XENOHALIOTIS californiensis occurs along the southwest coast of North America in California, USA, and Baja California, Mexico. However, because infected abalone have been transported to Chile, China, Chinese Taipei, Iceland, Ireland, Israel, Japan, Spain, Thailand, and possibly other countries, the geographic distribution of the etiologic agent is suspected to be wide where California Sarkai abalone, Haliotis rufescens, are reared or areas where native species have been exposed to swampy abalone. Mortality and morbidity. Sensitivity varies by species. Since the bacterium is known to cause the disease in black up to 99% mortality, white up to 100% mortality, in red up to 35% mortality, pink, also known as yellow, and green, also known as blue, abalone. Unlike other abalone species studied to date, the magnitude of abalone mortality is not well documented in pink and green abalone. However, in Baja California, Mexico, up to 100% of green, blue, abalone and 63% of pink, yellow, abalone can be infected, up to 43% of green fair and 71% of pink abalone with microscopic signs of disease. Degenerated or metaplastic digestive gland. The incubation period varies depending on the temperature but usually involves an extended pre-patency period of 3 to 7 months. Mortality usually occurs 1 to 2.5 months after the onset of visible clinical signs. XENOHALIOTIS californiensis has recently been observed, based on histological and molecular data, in several Asian countries including China, Chinese Taipei, and Thailand. Prevalence has not been well documented but up to 61% of H. Diversicolor supertexta have been infected on a farm in Thailand. However, like the European abalone, H. tuberculata, no abalone has shown clinical signs of wilt syndrome. Environmental factors. The disease, wasting syndrome, occurs at high water temperatures, 18 to 25 degrees Celsius, in abalone with moderate to severe infections. Clinical infections have been observed in H. Diversicolor supertexta elevated at 27 to 29 degrees Celsius. As abalone are obligate marine species, salinity tolerances of the rickettsia-like organism, RLO, were not investigated. Clinical and morphopathological signs. The disease, wilting syndrome, occurs at high water temperatures, 18 degrees Celsius and above, in abalone with moderate to severe infections. The incubation period of wasting syndrome is prolonged and generally varies between 3 and 7 months. Clinical disease is characterized by morphological changes in the digestive gland, which vary by species and may include degeneration, tubule atrophy, increased connective tissue, and inflammation, and/or metaplasia of the digestive tubules. Metaplasia involves the replacement of terminal secretory/absorbent acini with absorption/transport channels similar in appearance to the post-esophagus. Morphological changes are clinical accompanied by anorexia, depletion of glycogen stores, followed by the use of the foot muscle as a source of energy and subsequent death. The foot of affected abalone contains fewer and fewer organized muscle bundles, abundant connective tissue, and may contain more serous cells than unaffected individuals. Surviving abalone appear to remain infected, even in environments with low water temperatures, such as Northern California. Clinical biochemistry. A standard glycogen test can be used to test for glycogen loss in the digestive gland and pediosus muscle. However, A decrease in glycogen is linked to anorexia and the inability to digest food and is therefore not specific to this disease. Stained smears of digestive epithelia can be used to observe bacterial inclusions. However, examining small pieces of the posterior esophagus that have been dried on a slide and stained with a DNA fluorochrome be easier to interpret than stained smears. Microscopic pathology. The presence of oval basophilic intracytoplasmatic bacterial inclusions in the digestive epithelia, posterior esophagus, transport channels and metaplastic epithelia of the digestive gland and/or intestine. Metaplastic changes in the digestive gland that include the transformation of terminal secretory acini into absorbent/transport epithelia is amplified in abalone infected with X. Californiensis. Thus, the presence of X. Californiensis in the digestive epithelia in conjunction with the morphological changes noted above indicate the presence of a clinical wilting syndrome. Electron microscopy/cytopathology. Transmission electron microscopy, TEM, can be used to confirm the presence of RLO. However, this is not a confirmation for this agent due to the lack of unique morphological features. If used, TEM will reveal intracellular colonies of rod-shaped ribosome-rich prokaryotes with trilaminar cell walls in membrane-bound vacuoles in the cytoplasm of gastrointestinal epithelial cells. The rod to spherical dimorphic bacterium averages 332 × 1550 nanometers in the bacillus form and an average of 1405 nanometers in the spherical morphotype. The bacterium reproduces in intracytoplasmatic vacuoles. Polymerase chain reaction. A positive PCR amplification is only a presumptive diagnosis because it detects DNA and not necessarily a viable pathogen. Other techniques, preferably histology and ISH, should be used to visualize the pathogen. When used in conjunction with histology, PCR may be used for confirmation. Examination of the amplified sequence is recommended when examining a new host species or geographic area. In situ hybridization. ISH is the method of choice to confirm identification because it allows the visualization of a specific probe hybridized to the target organism. However, DNA probes must be carefully tested for specificity and validated in comparative studies before they can be used for confirmatory identification. ISH was developed to detect rickettsial-like prokaryotes in tissue sections. Ranking of tests against the purpose of use. The currently available methods for the monitoring, detection, and diagnosis of X. Californiensis are listed in the table below. A. For valuable broodstock, fecal polymerase chain reaction, PCR, can be used as the first screening and, if negative, the biological assay method can be used in combination with histology. B. Tissue impressions should be used in combination with PCR and possibly sequencing to confirm the agent. C. In new cases, such as a new geographic location, PCR and sequencing are recommended to confirm the identity of the bacteria. D. PCR alone is not confirmatory, but when used in combination with histology, it can be considered confirmatory. Experiments indicate that the pathogen can be transmitted through the water column and that direct contact between infected and uninfected abalone is not necessary. Above normal temperatures appear to have a synergistic effect on the disease. Catharus Martinez et al. 2000, Moore et al. 2000a, Raimondi et al. 2002. The results of experiments conducted by Friedman and Moore indicate that individuals of H. craterhodii and H. rufescens, stored at high temperatures, 20 degrees Celsius and 18.5 degrees Celsius, respectively, had higher mortality, more severe signs of wasting syndrome, and more severe infections with rickettsial prokaryote than those kept in cooler waters, 13 degrees Celsius and 14 degrees Celsius, respectively. In addition, the recovery of H. craterhodii affected by mass dieback deaths seems to be closely linked to temperatures. In affected livestock facilities, the severity of the disease may be limited if water temperatures can be reduced to about 15 degrees Celsius or less. Results from subsequent long-term, 447 days, Experiments using fed and hungry abalone indicated that the high levels of morbidity and mortality displayed by infected abalone were a consequence of disease and not direct heat stress. However, Rogers Bennett suggests that temperature needs to be explicitly included at least in management and recovery planning for H. rufescens. Oceanographic factors that lead to increased seawater temperatures, such as El Niño Southern Oscillation, have had a very negative impact on the recovery of H. craterodii in Southern California. These high temperatures were also associated with a dramatic increase in the number of H. rufescens with foot atrophy syndrome in breeding facilities in California. Despite the devastation that affected the populations of H. craterodii, a few large and older individuals can still be found, while a few small juveniles have been observed. In addition, the study by Tissot suggests that Atlantic abalone populations in Southern California may recover with weakening oceanographic conditions associated with the El Niño Southern Oscillation. The genetic structure of H. craterodii in the California Islands and on the coast of Central California has been assessed for recruitment patterns in surviving populations. Some populations that have faced catastrophic declines caused by wasting syndrome have developed resistance to the disease. Treatment: intramuscular injection and oral administration of an antibiotic were effective in reducing losses of infected abalone, and tissue retention of this therapeutic agent in the digestive glands remained elevated for an extended period of time and was still detectable. However, other antimicrobials had no measurable effect on disease. Using histology-based metabolomics and nuclear magnetic resonance spectrometry to determine that oxytetracycline administered in medicated feeds eradicated rickettsia-like wasting syndrome, thereby reducing metabolic degradation associated with the syndrome in elevated seawater by approximately 17 degrees Celsius. Disinfection: exposure to seawater containing rickettsia-like organisms at a concentration of more than 10 mg per liter, ppm, of calcium hypochlorite and disinfection of the equipment in a 1% iodophore freshwater bath for 1 hour were effective disinfection methods. In ranculture, the practice of frog breeding, for scientific purposes and for culinary purposes, is practiced in many European countries, including the United States. As with aquaculture, most of the challenges of ranaculture are related to livestock and disease. In aquaculture, iridovirus infections are reportable. Infection with frog virus 3, FE3, the type species of the genus RANAVIRUS, results in edema, hemorrhage and necrosis of lymphoid tissues, hematopoietic tissues, liver, spleen and renal tubules. Mortality rates in free-ranging amphibians are more than 90%. Preferred scientific name: Ranavirus infection in amphibians. Etiological agent: family Iridoviridae, genus Ranavirus. Ranaviruses are the agents of systemic infections of fish, amphibians and reptiles. The frog virus 3 is a species of ranavirus with a majority of strains originating from amphibians, susceptible host species. Natural ranavirus infections are known in most of the major Anura and Chordata families. Susceptible stages of the host. Susceptible host stages are all age classes, larvae, shapeshifters, and adults. Mechanisms of transmission. Ranavirus infections can occur through animal-to-animal contact, through ingestion of infected, dying, or dead individuals. Viruses can also spread between widely separated river systems and reservoirs. Transmission is supposed to occur by means other than water. Mechanisms include translocation of live fish or bait by recreational fishers. Prevalence. Ranavirus infections have been reported on 5 continents, including Asia. Its prevalence, based on intensive and generalized serosurveillance, the detection of antigens, is not known. Vectors. Amphibians can be infected in the same way as fish, and as such, details associated with EHNV are included here. Possible vectors include nets, boats and other equipment, or amphibians used as bait by recreational fishermen. Birds are potential mechanical vectors, as ranaviruses can be carried in the intestine, on feathers, legs and beaks. It should be noted that ranaviruses are likely to be inactivated at typical avian body temperatures, 40 to 44 degrees Celsius. Nevertheless, it is possible that ranaviruses, as evidenced by EHNV, can be spread by regurgitation of ingested material within a few hours of feeding. Ranaviruses have been recovered from free or farmed frogs, healthy or diseased, in America, continental Europe, the United Kingdom and Asia, and diseased free-living spotted salamanders Ambystoma maculatum in North America. Mortality and morbidity vary from species to species. Laboratory infections and field data show that mortality can range from low 0% to 100% of infected animals in an experimental group depending on the species, the virus and the age and health status of the host. Following short infection times. Environmental factors.
Natural epizootics of amphibian ranaviruses appear to be similar for aquatic ranaviruses, e.g., EHNV.
Epizootics appear to be seasonal and may be related to poor breeding, captive populations, and overcrowding, wild and captive. It has been hypothesized that for some amphibians such as salamanders, references, the disease is related to the annual occurrence of large numbers of young non-immune animals and their subsequent exposure to the virus in shallow waters. The gross pathology of ranaviruses appears to be similar to that associated with bacterial sepsis. Since opportunistic or resident bacteria can often be cultured from dead or sick frogs, it appears that many ranavirus outbreaks have been misdiagnosed as sepsis. Frogvirus 3, FE3, has been experimentally shown to cause edema, necrosis, hemorrhage, and death in recent embryos, tadpoles, and metamorphs. During experimental infections, metamorphic toads developed hemorrhages, accompanied by massive edema, to the ventral skeletal musculature, stomach and intestines. In addition, tadpoles of the Italian agile frog, Rana latistae, exposed to FE3 have occasionally become emaciated. Embryo mortality can occur 3 to 12 days after exposure with 40 to 70% tadpole survival. Clinical signs include depigmentation, peeling of the skin, and curvature of the spine. In general, lesions caused by FE3 appear to be milder than those caused by tadpole edema. Tadpole edema virus, molecular evidence suggests that VTE is a strain of FE3. The acute fatal disease has been replicated with experimental transmission to tadpoles and adults, although pathogenicity has varied among host species. Terminal clinical changes in tadpoles are obvious edema, especially in the ventral regions, and hemorrhages along the body. Internally, hemorrhages and necrosis usually extend to the stomach, intestines, and skeletal muscle, and may include the mesonephros, bladder, liver, lungs, and spleen. The highest titers of the virus occur in the stomach. Ranavirus infection in amphibians, main symptoms of ranavirus in sick Chinese giant salamanders, Andreas davidianus. The hyperemic and edematous orbit, A. Ecchymotic oral mucosa, B. Edema and skin ulcer, C. Congestion and swelling of the liver, Li, and spleen, SP, as well as purplish bruising in the kidneys, Ki, lung, Lu, intestines, IN, and bladder, BL, D, are shown. Ranavirus infection in amphibians. This slide shows a transmission electron micrograph of ranaviruses congregating near the cell border where they can be seen leaving the cell via a process called budding. Wood frog adults euthanized 2 days after infection with frogvirus 3. Figure 1. Diagram showing the location of foci of necrosis and or hemorrhage, red, inflammation, blue, and negative, gray, or positive, green, detection of ranavirus DNA with polymerase chain reaction assays of the liver, spleen, kidneys, femoral bone marrow, and skin. Figures 2 to 4. Skin, figures. 2a, small focus of mononuclear inflammation without detectable ranavirus staining among prominent dermal glands characteristic of anuran skin. Immunohistochemistry for epizootic hematopoietic necrosis virus. 2b, 3, mononuclear inflammation and 4, petechial hemorrhage. Hematoxylin and eosin, HEO. Figures 5 to 6. Kidney. 5. Focus of interstitial hemorrhage. 6. Higher magnification reveals slight necrosis of hematopoietic cells and minimal segmental degeneration of the tubular epithelium. Adult wood frog euthanized 4 days after infection of frog virus 3. Figure 7. Diagram showing the location of foci of necrosis and/or hemorrhage, red, inflammation, blue, and negative, gray, or positive, green, detection of ranavirus DNA with polymerase chain reaction testing of the liver, spleen, kidneys, nasal and oral mucous membranes, lungs, bladder, and skin. Figure 8. Lung. Mononuclear inflammatory cells in the ciliated mucosal epithelium above a bundle of smooth muscle, without any signs of necrosis. Hematoxylin and eosin, HEO. Figure 9. Kidney, slight interstitial edema. Figure 10. Lung. Positive staining for ranavirus in the ciliated epithelium infiltrated by mononuclear cells. Immunohistochemistry of epizootic hematopoietic necrosis virus. Figure 11. Urinary bladder. Mononuclear infiltrates, probably lymphocytic in the mucosa. Figure 12. Nasal cavity. Mononuclear infiltrates in the mucosa and submucosa, without obvious necrotic changes. Adult wood frogs euthanized 9 days after infection of frog virus 3. Figure 13. Diagram showing the location of foci of DNA ranavirus necrosis and/or hemorrhage, red, inflammation, blue, and negative, gray, or positive, green, detection with polymerase chain reaction testing of the liver, spleen, kidneys, nasal and oral mucous membranes, tongue, lungs, gastrointestinal tract, and skin. Figure 14. Skin. Figures. A. Focus of mononuclear inflammation in the dermis, hematoxylin and eosin, He, with B. Positive staining with ranavirus, immunohistochemistry of epizootic hematopoietic necrosis virus, ENG. Figures 15 to 16. Nasal mucosa. 15a. Mononuclear inflammation and 16. Small necrotic focus. Eel. 15b. The endothelium and necrotic focus are positive for ranavirus staining. ENG. Figure 17. Tongue. A small hemorrhage and mild mononuclear inflammation in the muscle layer and ventral surface epithelium. He. B. Positive staining of ranavirus associated with hemorrhage and inflammation of the ventral surface. IHC anti-EHNV. Figure 18. Lung, ciliated epithelium. A. Single degenerate cell, arrow, in an otherwise commonplace epithelium. He. B. Strong staining for ranavirus, arrow, which may be distinct from weaker nonspecific staining of smooth muscle, asterisk, is present in the epithelial region. EHNV-IHC. Current routine techniques for diagnosing ranaviruses in amphibians include immunohistochemistry, viral isolation from tissues and cell cultures, and ELISA and PCR. Sublevel or clinical infections can only be detectable by cell culture or PCR. Definition of the suspected case: apparently healthy, moribund or dead amphibian whose skin and/or parenchymal tissues show histological signs of focal, multifocal or locally extensive liquefactive or coagulant necrosis with or without intracytoplasmic basophil inclusion bodies. Confirmed case definition. Amphibian, apparently healthy, moribund or dead whose skin and/or parenchymal tissues contain histological signs of focal, multifocal or locally extensive liquefactive or coagulant necrosis with or without intracytoplasmic basophilic inclusion bodies and/or in which ranavirus is evidenced by the following means. 1. The characteristic EPC in cell culture and cell culture are positive for ranavirus in the immunoperoxidase assay or ELISA or PCR for antigen capture, or 2. Tissues positive in antigen capture ELISA or immunoperoxidase staining or immunoelectron microscopy or PCR. And for 1 and 2, where PCR is used, The sequence compatible with the RANAVIRUS is demonstrated by PCR-RIA or PCR sequencing, the main shrimp culture area's global map. American foulbrood is a disease regulated in the European Union in the context of trade and international trade. It is classified as a Category 1 health hazard in France and EU countries. Colony damage, AFB, is a brood disease caused by the spore-forming bacterium Pinobacillus larvae. Only young larvae are susceptible to infection, adult bees are not affected. When a larva is infected, the entire brood of the colony can be quickly affected by the bacteria. The colony will then not be able to raise a sufficient number of young workers, which will lead to its weakening or even death. Young larvae become infected by ingestion of P. larvae spores. Life cycle: the ingested spores germinate, proliferate in the larva's intestine and then pass through the epithelium of its digestive tract, inducing its death. If the dead larva is not eliminated through the hygienic behavior of the cleaners, the infected tissue disintegrates in the capped cells. Subsequently, the remains of the larva dry out, producing dark, hard scales, which are very difficult for the workers to clean. These scales can contain up to 2.5 million spores, which are highly infectious to other larvae. This can lead to rapid spread of the disease if not detected in time. P. larvae spores are highly resistant to disinfection and can survive for up to 30 years and more. Criteria for suspicion and/or consequences of infection for the colony: clinical signs: sagging opercular, concave lids, abnormally dark in color, abnormal perforations of the lids, presence of small holes, pads with a wet/oily appearance, larvae brown in color and stringy consistency. The match test is used to assess the viscosity of the larva, B and C. After this stage, the larva dries out and forms a hard scale, which adheres to the cell wall. Presence of a pupal tongue, very characteristic sign but rarely observed. D. Irregular brood with a mosaic appearance, resulting from the hygienic behavior of cleaner bees which remove dead larvae from operculated and uncapped cells. E. Foul odor specific to diseased larvae. Weakened colony, decreased activity on the flight board. Criteria for suspicion and/or consequences of infection for the colony: clinical signs: sagging opercular, concave lids, abnormally dark in color, abnormal perforations of the lids, presence of small holes, pads with a wet/oily appearance, larvae brown in color and stringy consistency. The match test is used to assess the viscosity of the larva, B and C. After this stage, the larva dries out and forms a hard scale, which adheres to the cell wall. Presence of a pupal tongue, very characteristic sign but rarely observed. D. Irregular brood with a mosaic appearance, resulting from the hygienic behavior of cleaner bees which remove dead larvae from operculated and uncapped cells. E. Foul odor specific to diseased larvae. Weakened colony, decreased activity on the flight board. How to inspect the hive. Review frames individually. Examine the brood, especially the capped cells, for abnormalities described above. If dead brood is observed, check the viscosity of the larvae with the match test. Prevention/treatment. Prevention. Attention should be paid to the prevention of the disease, which is based on the application of good beekeeping practices and prophylaxis.
The introduction of bees, e.g., nucleates, queens, collected swarms, hive products, honey, pollen, wax, etc., and equipment into a colony or apiary must be carried out with great care.
Looting must be avoided. All colonies, and in particular the brood, must be regularly inspected in order to detect any abnormalities early. Brood frames must be renewed regularly. Treatment. In the event of an epizootic, the health measures applied aim to isolate and eradicate the disease. A protection zone is established around the outbreak and all movements of bee colonies and beekeeping equipment are prohibited. Heavily affected colonies are destroyed to prevent the spread of the disease. There are several ways to destroy colonies. Generally, the hives are closed in the evening, when all the bees are inside, and then euthanized, using sulfur dioxide, for example. The hives, bees and beekeeping equipment are then incinerated. Antibiotics are not effective on spores, which are highly resistant. There is no veterinary medicine authorized for the treatment of American foulbrood in the European Union. What to do in case of suspicion? In the European Union and France, American foulbrood is a regulated disease. In case of suspicion, the departmental directorate in charge of the protection of populations must be alerted. Samples of the suspect brood should be sent as soon as possible to an approved laboratory for confirmatory analysis. European foulbrood is a common brood disease of bees. It has a low prevalence in most European Union countries, with some exceptions in certain regions where the disease is problematic. In the EU, European foulbrood is a disease for which national programs can be recognized. Damages to colonies. European foulbrood is a brood disease caused by the gram-positive bacterium Melissococcus plutonius. The larvae usually die from infection at 4 to 5 days of age, mainly before capping, but can die from infection at any age. Adult bees can carry the bacteria, but it has no known harmful effects for them. Colonies with European foulbrood can recover, but sometimes most of the brood is affected, causing the colony to weaken and die. The damage caused by the disease appears to vary drastically across regions and countries. Life cycle. Young bee larvae, generally less than 48 hours old, become infected by ingesting M. plutonius bacteria via contaminated food. The bacteria multiply in the gut of the larvae, which can sometimes be expelled from the colony by nurses. Those that die from the infection can be colonized by secondary bacterial agents present in the hive such as Pinobacillus alvei, Enterococcus faecalis and Brevibacillus laterosporus. Some larvae die after capping and fail to reach the pupae stage, emptying their M. plutonius infected feces into the socket. Others manage to transform into nymphs and become adults but leave behind infectious material. Although it is a bacterium that does not form spores, M. plutonius can survive in harsh conditions and remain infectious for several years on the wax. However, it is much less resistant to disinfection than spore-producing bacteria such as Pinobacillus larvae, the causative agent of AFB. Criteria for suspicion and/or consequences of infection for the colony. Clinical signs: irregular brood, mosaic, withhold, collapsed opercula. Change in color of infected larvae, from pearly white to pale yellow, often accompanied by a loss of segmentation, Figure 1 and Figure 2. Infected larvae may then turn a brown and grayish-black color, sometimes leaving a dark brown scale, Figure 2, which is easily detached of the cell, unlike the scale due to American foulbrood. Larvae die mainly in non-capped cells, open brood, but sometimes in capped cells, closed brood, Figure 2. Dead larvae take on a translucent appearance, revealing their tracheal system. Dead larvae are often in an abnormal position, twisted or in an upright position, Figure 2. Dead brood sometimes gives off a foul or sour smell. How to inspect the hive? A diagnosis based only on visual clinical examination is not reliable. Laboratory confirmation is necessary. Inspect each brood frame individually. The brood, including capped brood, to check for suspicious clinical signs. Assess the viscosity of dead larvae using the matchstick test for differential diagnosis with American foulbrood. Larvae affected by European foulbrood are rarely stringy. If they are, the string is less than 1 cm, Figure 3. Figure 3, matchstick test on larvae affected by European foulbrood, A, or American foulbrood, B, handles. The focus should be on preventing the disease, apply good beekeeping practices, good varroa control and good prophylaxis.
Exercise caution when introducing bees, e.g., buying nuclei, retrieving swarms of hive products and material of unknown origin.
Prevent, avoid looting. Inspect colonies frequently, especially brood. Renew the wax regularly. 1/3 of the wax should be replaced each year. Ensure that there is always enough food in the colony and feed as needed. Regularly check the appearance of the brood, report abnormal symptoms immediately to the apiary inspector. Eliminate weak colonies. Avoid hypothermia of brood frames. Colonies of great vitality with young queens and good cleansing instinct. Ensure that there is always enough food available for the bees. No feeding with honey from outside the farm. Controlling varroa mites according to the concept, www. Obey. /varroa. Regularly renew frames. Caution when exchanging frames between different colonies. Location with good honey flow conditions. When purchasing colonies, ask for written confirmation of the bee's state of health by an official, apiary inspector. Do not place colonies near sequestration areas. Thoroughly clean and disinfect used equipment before first use. There is currently no cure for colonies affected by European foulbrood, and these colonies are monitored by the apiary inspector. In case of an outbreak, eliminate severely affected colonies using one or more of the following methods. Seal the hive entrance and any openings at night when the bees are inside. Euthanize the bees by burning a sulfur wick inside the hive. Carefully remove the frames from the hive. Clean wooden materials with a hot water solution containing 3 to 5% caustic soda, sodium hydroxide, and disinfect them with a blowtorch. Pack dead bees and infected frames to prevent access by other bees, and dispose of them as quickly as possible, ideally by incineration. For colonies with mild infection, use the decanting method, creating naked swarms, by destroying all frames, including brood, while preserving only the adult bees. Regular inspections of colonies are necessary to detect any new outbreaks early. Disinfect any equipment that has been in contact with infected colonies, such as frame lifters. Destroy weak colonies, as they are more vulnerable to infection. What to do if you suspect an outbreak? European foulbrood is not classified as a notifiable disease within the European Union, resulting in minimal issues in some areas. While control measures are not mandatory at the EU level, certain national programs may be recognized by individual member states, as per Council Directive 92/65/EC.